Hello, everyone. Welcome back to Science in the Age of COVID-19. Today, we are excited to welcome Dr. Tyler Starr to our series. Tyler is a postdoctoral fellow in Dr. Jesse Bloom's lab at the Fred Hutchinson Cancer Research Center. For those of you who've been to our talks, you've probably heard from Jesse before. A bit about Taylor. Taylor is a biochemist by training and uses a combination of computational approaches and biochemical techniques to predict the effect of mutations on the function of antibodies. Since the onset of the pandemic, Taylor has applied this technique to highlight the functional diversity among antibodies against the receptor-binding domain, or RBD, of SARS-CoV-2. We'll hear about all that and much more today in his talk. So Taylor, welcome. Thanks for your time. And we look forward to hearing from you.
Awesome. Thank you so much for the introduction. And thanks everybody for joining. All right. And so, as was just mentioned today, I'm going to tell you all about some of the work we've been doing to explore functional diversity among SARS-CoV-2 antibodies, kind of highlighting one of our recent papers, which looks at some principles for how to maximize breadth and resistance to escape among antibodies to SARS-CoV-2. I can get a laser pointer. There we go. And as I get started, I just want to really highlight two, I'll highlight a bunch of collaborators throughout, but in particular, I want to highlight my advisor, Jesse, as well as a graduate student in the lab, Allie, who have really been, full key collaborators on all of the stuff we've been doing in this realm. And it's been super fun. All right, so just as a general introduction, I'm going to tell you all about SARS-CoV-2 spike, which is a protein that we're all deeply familiar with, and hopefully at this point, many of us are immunologically familiar with it at this point as well. But spike is the trimeric glycoprotein that decorates the surface of SARS-CoV-2 virions. And it's the protein on the surface that facilitates entry of this virus into host cells. It does this by using its receptor-binding domain, or RBD, to bind to ACE2 receptors on host cell surfaces. And this binding between RBD and ACE2 receptor triggers further conformational changes in spike, which unleash the fusion machinery to enable fusion of the viral and host membranes, which is what facilitates the initiation of cellular infection. So as the viral entry protein, spike is, of course, a primary target of neutralizing antibodies, including those elicited by infection, but also, of course, via the vaccines for which spike is the sole component of the virus needed to protect against infection. And so at this point, we know there's diverse epitopes on the spike protein in general that neutralizing antibodies can target. But antibodies to the RBD in particular have proven incredibly important. And so some of the evidence for the importance of RBD antibodies includes the fact that the majority of neutralizing activity in polyclonal sera derives from RBD-directed antibodies. And so what I mean by that is if you take RBD protein, isolated protein, to pull out RBD-specific antibodies and then measure how much neutralizing activity is left in a sera sample, you can see that the majority of neutralizing activity in most individuals is pulled down by the RBD specific pull down, meaning that the majority of that potent neutralizing activity of sera is directed against the RBD. Furthermore, the RBD is a target of all of the monoclonal antibody drugs that currently have emergency use authorization, as well as most of the monoclonal antibodies that are in development across various stages of clinical trials. And though, even though we have these incredibly successful vaccines, it's important to note that monoclonal antibodies are still going to be incredibly important in this virus for the long-term future, for example, in individuals who can't mount vaccine responses. Antibodies to spike in general and the RBD in particular have various mechanisms of action or mechanisms by which they neutralize virus. Kind of the simplest mechanism by which antibodies neutralize SARS-CoV-2 is by directly binding to the same interface of the RBD that interacts with ACE2, and thereby just directly competing off ACE2 binding. And so many of the most potent antibodies, most potently neutralizing antibodies, simply just block ACE2 binding. But there's some other important mechanisms as well. One sort of neat mechanism that was unveiled, just prior to the pandemic in 2019 by Lexi Walls and David Wiesler, is that antibodies can sort of prematurely trigger the same type of refolding cascade that ACE2 binding normally initiates. And so this is just shown here that, normally the spike protein, when it binds to ACE2, converts to this post-fusion form, which if it's close to a membrane, that's what will, enable the juxtaposition of viral and host membranes to enable cell entry. However, an antibody can trigger the same type of conversion to the post-fusion form, far away from a host cell membrane. And so then you end up with a virus that's just coated in these post-fusion sort of now inactive spike proteins. And so that virus is thereby neutralized. Antibodies can kind of do the opposite. They can also just simply prevent this refolding to the post-fusion conformation. So not directly by competing with ACE2 binding, but just preventing those downstream conformational changes. And then, of course, there's tons of FC-mediated effector functions that these antibodies can elicit in order to confer in vivo protection. Then finally, I want to highlight, and as I'll show with some graphics later on in the talk, there's quite a diversity of epitopes in spike in general, but even within this small RBD protein, there's a lot of different RBD epitopes. And so one of the goals of our work is to sort of understand how these epitopes differ in their functional properties and some of their, evolutionarily relevant phenotypes. And so to kind of motivate that a little bit more, kind of the key question that a lot of the work I've been working on is trying to address is this question that's been crystallizing over time in terms of how this SARS-CoV-2 virus is going to be evolving antigenically in the short and perhaps medium to long term. And so I think these days there's not really much debate left about whether this virus will evolve antigenically, and it's much more about how it will do so. But even back in the summer or last spring, when global variation among SARS-CoV-2 was low, we were already seeing some hints that this was perhaps a virus that was going to be able to evolve antigenically. And so this insight initially came from our first study on the SARS-CoV-2 RBD. And in this study, we performed a so-called deep mutational scan in which we measured the effects of all amino acid mutations in the RBD on the affinity for binding to human ACE2 receptor. And so this is just a snippet of that data where I'm zooming in just on the SARS-CoV-2 positions that actually contact ACE2 receptor. And then the heat map shows the effect of each of the 20 amino acid changes that can be introduced at each of these positions, where white squares indicate mutations that are perfectly well tolerated with respect to receptor binding. And blue actually indicates mutations like Y453F or N501Y, which actually enhance receptor affinity. And so one of the kind of key details we saw in this study was that there was actually a pretty remarkable degree of mutational tolerance within the RBD, even among those positions that putatively should be among the most constrained, those that are actually directly responsible for binding to receptor. And that suggests that there's kind of a porous evolutionary space over which this virus can evolve. To kind of contextualize what that might mean for antibody binding, I'm showing here kind of the average constraint on ACE2 contact positions mapped to the RBD structure, where I take a site and I color it according to the average effect of mutations at that site on binding. So again, white indicates sites where mutations on average don't really impact affinity, whereas red are the positions that are more constrained, where mutations on average would be deleterious with respect to receptor binding. And so among the ACE2 contact positions, you can see there are some that are pretty well constrained. Then there's also this sprinkling of much more mutationally tolerant positions. In terms of what that can mean for antibody escape, this is kind of where we got our first initial hints, was this early study from Baum et al., so scientists at Regeneron, who took wild type SARS-CoV-2 virus and grew it in the presence of some of their monoclonal antibodies, and then sequenced the sort of escape variants that came out from this little in vitro evolutionary scenario. And these antibodies were among those potently neutralizing antibodies, like I mentioned, that target the same surface that contacts ACE2. But what we could see was that the sites that were facilitating escape were sort of cherry picking out these mutationally tolerant sites from this sort of hodgepodge of tolerant and constrained positions, really emphasizing that, any degree of mutational pliability is perhaps sufficient to enable this type of evolutionary escape from antibody binding. So, as the pandemic has progressed more and more, as I said, we've been seeing these variants emerge across the globe with a lot of these mutations at these, sites that are targeted by some of these super potent antibodies. And it's led to, I think many times over this question being asked, oh, here's a new variant. Like, what does this mean for us? In large part, these answers have been coming from a lot of scrambling from a lot of scientists to try to quickly understand the impact of novel mutations in this virus, and particularly in this domain. And so the goal of the work we've been doing is in part to fast forward some of this scrambling and prospectively map how new mutations might impact key phenotypes like antibody binding, to kind of prioritize what new mutants might be of phenotypic importance. And then, as I'll talk about at the end of the talk, we also kind of use this idea to see kinds of patterns that can help inform future antibody and vaccine development. So just a quick outline of what I'm going to go over today. I'll first tell you about the method we've been using to completely and prospectively map antibody escape mutations. I'll tell you really briefly about a couple of themes and trends we saw when we mapped how mutations enable escape from some of the monoclonal antibodies and cocktails that are approved for therapeutic use. And then I'll end with one of our more recent studies that begins to look at some of the principles that might enable maximization of robustness to escape among SARS-CoV-2 antibodies. All right, so our method for this kind of complete mapping of escape from antibody binding is leveraging the platform of yeast surface display, which is where we did that first demutational scan for receptor binding affinity that I showed the snippet of. And so yeast surface display is a classic platform for sort of high throughput biochemistry in different contexts. But the way we're using it is to express soluble RBD protein on the yeast cell surface. And when it's there, we can use various fluorescent metrics to measure biochemical properties of the yeast displayed RBD protein. In particular to this work, we can incubate with antibodies that target the RBD and detect that antibody binding via fluorescence. And so if we just take, yeast that are all expressing the wild type SARS-CoV-2 RBD and we incubate with an RBD binding antibody, we get a flow cytometry cloud that looks like this, where we have antibody binding on the y-axis and the degree of per cell RBD expression on the x-axis, and we get this nice cloud of bound RBD expressing yeast. And so to kind of use this platform to map how mutations impact antibody binding, we start by using instead of sort of a wild type population of yeast, we're using a library in which each yeast is expressing a unique RBD mutant on its cell surface. And this library that we've constructed has virtually all of the possible amino acid mutations that can be introduced as single mutations within this 201 amino acid domain. So then, similar to as I showed here, we just incubate with antibody, and then we can look at sort of the cloud again via flow cytometry. And because some of the mutations in this library escape antibody binding, instead of the tight cloud, we do get a bunch of bound cells, but then we have cells that begin to drop off in the levels of antibody binding, reflecting again the presence of mutations that escape that binding. And so all we have to do is use FACS to sort out these antibody escape cells. We use deep sequencing to quantify the frequency of each mutant in sort of the pre-selection library compared to this antibody escape enriched population. And then we just quantify escape by looking at the fraction of pre-selection cells that fell into the antibody escape bin. And so the way I'll generally show this data is in these logo plots, where we can show different sites in the RBD, and then the height of a letter indicates the degree to which that particular mutation at that site facilitates escape. And in particular, we use this metric called the escape fraction, which in its most direct form means the fraction of times a yeast of a given mutant was found in the antibody escape bin compared to its overall population frequency. So just to hammer that home, something like this orange mutant, had 4 reads pre-selection, and all 4 of those cells fell into antibody escape bin. So it has the maximum fraction escape of 1. All right, I'll go over just a couple examples of how this data looks and how it makes sense to us from what we know about biochemistry and evolution. So what I'm showing here is one of the early antibodies we mapped, the CR3022 antibody. And what I'm showing here is sort of the complete data. So this is a line plot for all sites in the RBD And then the y-axis is the amount of escape we measure at that site. And so you can see, of course, some sites enable escape from this antibody, but lots of sites don't. We can then construct these logo plots, like I said, zooming in on the sites that do have large effect escape mutations and see the particular amino acid changes that are facilitating this escape. And then last is, further proof of principle, we can map the escape to the actual RBD structure here, where we, project sites from white to red, where red indicates the sites that had the strongest degree of escape from binding to this antibody. And so we can see we get a little epitope patch down here, which when we compare to the CR3022 bound crystal structure, we see maps bang onto that interface as you'd expect. And so with this type of deep mutational scanning data, we can recapitulate details about the actual structural footprint or the structural epitope of the antibody. But beyond that, we also get some knowledge of the actual functional hotspot nature of this RBD antibody interaction. The last little thing I want to point out is that we can actually use these binding escape maps to, perhaps we can say predict or at least make sense of the types of mutations that actually emerge when this virus is under selection by particular antibodies. And so this is an example of an antibody CoV-2 2499 and the escape map we get for mutations that facilitate escape from binding by this antibody. In parallel, Pavlo Gilchuk and Seth Zost from James Crow's group performed these types of viral escape selection experiments once again, where you grow antibody or virus in the context of antibody and then sequence the mutants that escape that antibody. And so when they did this, 16 different times, 5 times they saw viral escape mutants emerge. And in particular, they saw multiple counts of these 2 particular mutations, so G4460 and Q498R. And so first of all, we can look at our map and say, great, those mutations are in our maps. We did indeed see these mutations facilitate escape from binding. But at the same time, we can also see, we have, 30 or 40 different mutations in here. So like, what was the actual predictive power here? One of kind of the first pass things that helps make a little more sense of what actually happens in this evolutionary scenario is that we can facet on just amino acid changes that are accessible via single nucleotide changes, because that's the type of change you're really going to be selecting for in this short-term evolutionary scenario. And so now we can see, okay, these 2 mutations are 2 out of maybe a dozen or so mutations that should enable escape from antibody binding. And sort of the last detail that we can layer on to this data is sort of other information about these mutations, notably what their consequences are for things like receptor binding. And so the way we typically show these escape maps is that, it's logo plots as I showed, but then we can also color the mutations by their sort of side effects on things like ACE2 binding, where mutations that are perfectly compatible with ACE2 binding stay this dark brown color, and they sort of fade away to this more transparent yellow when they have more and more deleterious effects on ACE2 binding. And so when we do that, finally, we can see that really the 2 mutations that came out from this selection are the 2 that enable large effect escape from this antibody without having deleterious consequences for ACE2 binding. So kind of when we did this, many times with many different antibodies, we saw that in general, we can make sense of what mutations emerge from this simple set of prospective mapping of how mutations impact antibody binding and receptor binding. All right. So with that in hand, I'll go over just briefly some of the themes and trends we've seen when we explored how mutations enable escape from some of the antibodies used in the therapeutic treatment for SARS-CoV-2. And so going back to, I think, first in November, the FDA began issuing EUAs for some of these monoclonal antibodies and their cocktails. And so we now have 4 different monoclonal antibodies in the context of 2 different cocktails that are, approved for therapeutic usage. And they've shown, a lot of promise in reducing viral loads and reducing incidence of severe infection when administered properly. There's a lot of trials about how they can prophylactically protect via passive immunization. And so really these antibodies are proving to be a really important drug class for the treatment of SARS-CoV-2, but really highlighting how they're also a very tenuous class. Even in the short months since November, there's already been a striking of an EUA of one of the monoclonal antibodies, LY-CoV-555 or bamlanivimab, for concern about how it might be escaped from some of these emerging variants of concern. And so to see kind of why that's the case for this monoclonal antibody, we can turn simply to our prospective escape map that we generated for 555. where we see, a handful of mutations at some of these mutationally tolerant sites given by their brown colors that enable escape from 555 binding. And so, if you've been paying attention to a lot of the news on these variants, you're going to be seeing a couple sites crop up in this profile that would, really make your radar start buzzing, including E484 and L452. And so indeed, we can see in these maps two mutations, L452R and E484K, which have been emerging again and again in parallel across multiple of these variants of concern. And so this kind of explains why bamlanivimab sort of, it was the first to get through. It was developed incredibly quickly. But as SARS-CoV-2 began to evolve more of these variants globally, it became pretty apparent that over time, a lot of these mutations that render 555 inactive are going to be accumulating. And so obviously, I think an important question is why mutations that escape this antibody are so prominent. So it's, of course, the usage of this antibody in particular is not so widespread that it's driving this rise in frequency of mutations like E484K and L452R. But instead, we can get some insight from a recent study from the lab led by Ali Greeny, who compared maps of escape from individual antibodies of diverse RBD epitope classes to the types of escape mutants that you get when you do this mapping approach with polyclonal sera, either from infected individuals or vaccinated individuals. And when she did this, she saw that mutational escape from polyclonal sera was largely driven by the types of mutations that escape a particular class of antibody, which are known as class 2 antibodies, of which LY-CoV555 is a representative. And so this suggests that antibodies like LY-CoV555 are sort of immunodominant in natural immunity. And so the same types of, population level immune pressures that are going to be driving SARS-CoV-2 evolution, now and into the future, are going to be fixing the types of mutations that escape this class of antibodies, which renders them pretty fragile as sort of a drug or a therapeutic class. And so, of course, the reason why Eli Lilly even requested that the EUA for bamlanivimab be revoked is because they really wanted to move toward this cocktail approach, which is, of course, a common approach for minimizing viral escape, for the simple reason that instead of requiring a single mutation to escape binding from a monoclonal antibody, if you have a cocktail of two antibodies, then you're going to need two or more mutations to escape the cocktail. And so, now Eli Lilly is providing 555 as a cocktail with this other antibody, LY-CoV016, which has an escape map, which highlights a bunch of different sites across the RBD in general. However, we can see some sort of low-level overlap in some sites where mutations escape binding from both antibodies. And so if we do combine these antibodies together as a one-to-one combination and then just see what mutations enable escape from their kind of dual cocktail, we do see some sort of biochemically dramatic mutations in these sites of structural and sequence overlap between these escape maps, where these individual mutations can facilitate escape from the two antibody cocktail. The reason for that can be seen pretty clearly structurally, which is the fact that these two different antibodies basically target different sides of this one little structural element, which we call the ACE2 binding ridge, such that some of the sites that are at the structural overlap between these two epitopes, kind of down the middle of this ridge here, can enable escape from both antibodies simultaneously. Kind of maybe the more important concern, I guess, at this point for some of these cocktails is that a lot of these SARS-CoV-2 variants that are already beginning to emerge are actually fixing multiple mutations at once. And so, despite the fact that there's single mutations that can escape the cocktail, there's also obviously dual combination mutations which can do so. And this includes combinations of mutations that are found in some of these, most prominent variants of concern, such as B1351 and P1, which combine E484K, which we already talked about escaping 555, but they also combine these K417 mutations, which is pretty much the most prominent site of escape for LY-CoV016. And so I think this just kind of highlights about why this overall mapping strategy is super important for sort of knowing the vulnerabilities of some of these monoclonal and cocktail therapeutics. And also it shows that really surveillance is going to continue to be extremely important in the administration of these drugs. So as some of these variants take over in locales, I think that has important implications for which antibody cocktail you might choose to turn to for usage. All right, I'm going to skip over some of our Regeneron cocktail results and kind of just summarize a little bit about what we saw from this work mapping escape from therapeutic antibodies. So in general, many of the first generation of antibodies that were just developed incredibly quickly, and it's super impressive. These antibodies were generally prioritized by their potent in vitro neutralization phenotypes. And so this type of discrete lead selection for potent in vitro neutralization leads to selection of this particular type of antibody that targets that ACE2 contact interface that I talked about in order to directly block ACE2 binding. Because these same epitopes that are potently neutralized by these ACE2 competitive antibodies are also highly targeted in sera, we think that there's this immunodominance at a population level that's driving perhaps variation at these same epitopes that are being targeted by these therapeutic antibodies. And so the question is sort of whether we can, diversify the repertoire of monoclonal antibodies that are developed and in doing that, can we identify epitopes and antibodies that are going to be more robust to ongoing viral evolution, such that we're not going to constantly be playing catch up with our antibodies as this virus continues to evolve over time. And so that brings me to the kind of the main part of the talk, which is talking about this new project we've been working on, looking into how we might be able to maximize robustness to escape among antibodies to the SARS-CoV-2 RBD. So to set the stage for this, I want to kind of emphasize again what I brought up before, which is that there are many different RBD epitopes. And so the way we see RBD epitope space is kind of what emerges from all of the different antibodies we've performed this viral escape selection on. And so these are all different antibodies that we've generated these escape maps for. And what we've done here is created this multidimensional scaling layout, which is sort of like a T-sign or a principal components analysis that lays out antibodies according to their similarity in their escape profiles. So two antibodies that are closer in this space have more sort of shared sites of escape compared to antibodies that are further away on this space. And so this diagram sort of partitions out all of the different epitopes we've seen in doing these antibody mapping experiments. To kind of give some context to this epitope space, we can dig in a little bit to the anatomy of the RBD. So these pie charts for each of the antibodies represent sort of the overall structural class of sites that escape that antibody, which we partition into these three categories. We have the core RBD, which is in orange, which is this part of the RBD that forms like the kind of folded beta sheet core of the RBD and is a little bit more distant from the ACE2 contact interface. In light blue, we have sites that are part of this sequence element called the receptor-binding motif, which is just a linear sequence element that contains all of the architecture for ACE2 binding. And then within this RBM, we have in dark blue the actual residues that are, direct ACE2 contact residues defined, as a 4-angstrom cutoff from ACE2 residues in crystal structures. And so that kind of gives you some insight into this space where we're seeing a lot of the core RBD epitopes are down here, whereas the sort of ACE2 contact and RBM surface is sort of tiling across from some of the ACE2 contact positions, maybe along the back of the RBM, back to ACE2 contact positions on the other side. We can see that as we begin to project our escape maps onto this structure from some of the representatives across this space. And so I'm showing here the four kind of classes of epitopes that are represented among the four antibodies that we currently have approved for therapeutic treatment of SARS-CoV-2. And so you can see 016 is sort of this front-facing kind of like this area here that incorporates a lot of the ACE2 contact residues and the RBM. We can see antibodies like Regeneron 10933 are then kind of popping up the top of this little residue of this little nub onto the F486 site. And then we can see something like LY-CoV555 is beginning to traverse to the back of this RBM. And then as we get to Regeneron 10997, we're kind of coming along this little backside here. And so you can see that, there are different epitopes represented amongst the four antibodies that we have approved for therapeutic treatment, but they're all sort of targeting, again, this receptor binding surface. And we have a lot more epitopes either within this surface as well as in a lot of components of this core RBD. And so we kind of flush out the rest of this diagram by laying out kind of the rest of antibodies that span this space to really, once again, flesh out the different epitopes we've seen. And so again, we can see the RBM space on the structures begins to kind of traverse over the side and the back of the top or the ACE2 contact surface of the RBD. But then we can also see a bunch of unique epitopes within the core RBD, such as S309, which is kind of down here on the backside of the RBD, or the set of antibodies here, which all target this sort of cryptic face of the RBD here in the front within the core RBD, or this unique antibody S2H97, which targets an epitope sort of down the side below antibodies like LYCOV016. And so this is just to emphasize that there's a lot of different epitopes out there, and it might be important to understand, how these different epitopes differ in things like robustness to viral escape, such that we can have a more diverse array of antibodies in our arsenal for therapeutic antibodies, but also just to understand the range of responses we can get from something like a vaccine. So for this question about how we can maximize robustness to viral escape, some of our first clues came from this antibody that's in therapeutic development called S309, which comes from Vir. So S309 is actually an antibody that was also quickly isolated at the beginning of this pandemic. And that's because it came from sera that was already in the freezer that came from a patient who had recovered from SARS-CoV-1. So, a genetically related but distinct virus that caused the 2002 to 4 epidemic. So, of course, we wouldn't be talking about this antibody except for the fact that it, through this epitope, it can actually cross-neutralize not only SARS-CoV-1, but SARS-CoV-2 itself. So it already points to some degree of breadth across this diverse lineage of SARS-related coronaviruses. S309 is interesting in that because of the epitope it targets, it's not actually as potent at in vitro neutralization of SARS-CoV-2 as some of these other antibodies that target the RBM or directly compete with ACE2 for binding. So when you do it in vitro neutralizations, you might have a little bit weaker, neutralization activity of this antibody, but it kind of makes up with that with a lot of effector functions and other mechanisms of action, such that when you actually put it in vivo, including in humans, you get just as good of protection from an antibody like S309 as you do from some of these antibodies that were instead prioritized for their neutralization capacity. And so some of the early numbers that have been coming out from the phase 3 trial in this antibody show that it's just as effective as something like LYCOV555. And then kind of the important kicker about this antibody is that in contrast to things like LYCOV555, S309 is currently being shown to retain activity against all of these SARS-CoV-2 variants of concern and a lot of the newly emerging variants. And in general, sequence variation within its epitope is quite rare. And so this kind of suggests that there may be some sort of relationship between the degree to which an antibody is broad at sort of the larger scale evolution of SARS-related coronaviruses over deeper evolutionary time, and kind of how that breadth translates to breadth within the ongoing variation of the SARS-CoV-2 pandemic itself. And this premise or this relationship between breadth at these two levels is also underscored by another clinical stage antibody from Adagio and Adimab, which looks more like these antibodies down here. It kind of targets this little surface here, which once again, these antibodies have some breadth across SARS-related coronaviruses, and they're also broad against emerging variants of concern. And so, in this study, what we sought to do was sort of leverage this observation of the relationship between breadth at the deep evolutionary scale and, neutralization of variants of concern to understand how these properties relate across this broader space of possible epitopes. And so we worked together with Vir on a panel of antibodies shown in red with the squares here that sort of span the diversity of epitopes represented in this epitope projection space. This panel of antibodies that we're using here includes some of these antibodies that are targeting relatively unique epitopes like S309 and S2H97, but it also includes epitopes, again, that sort of span this arc of epitopes along the RBM. And so we used this panel of antibodies to profile escape using our escape mapping approach that I've been using. And so here I'm showing you 3 of the antibodies that are all part of the RBM epitope group. And we can see here, the different sites that enable escape from these antibodies and compare that to the frequency of these escape mutations among sequences of SARS-CoV-2 that have been gathered across the world. And so, for example, you can see something like S2D106 is, selecting escape at E484, where there's a high degree of variation at that position, or something like S2X58 suffers from both E484 and L452, which is also a frequent mutation with L452R. And so we can simply just sum the frequency of all of these escape mutations of each of these antibodies across the GISAID database of sequences and see the degree to which each of these antibodies is susceptible to some of the ongoing variation in SARS-CoV-2 by just looking at the total frequency of these escape mutants. Then to kind of go along with that, we also extended our mapping approach to explore not only mutations within SARS-CoV-2 itself, but also to explore the greater breadth among SARS-related coronaviruses or sarbecaviruses. And so I'm showing here the overall evolutionary tree of SARS-related coronaviruses, of which we're intimately familiar with SARS-CoV-2 here, this human strain, as well as SARS-CoV-1, which, as I mentioned, is genetically distinct clade from SARS-CoV-2, but there's some relationship. But then there's also these other lineages from bats across Asia, Africa, and Europe, which span this entire sarbecavirus clade. And so what we did was, similar to how we made our mutant library, we also constructed a library that just includes all of these different sarbecavirus RBDs, and we performed the same type of antibody escape selection to determine the ability of each of these antibodies to bind the breadth of sarbecavirus RBDs. And so we can represent that data here in this type of heat map, where each column is one of the antibodies that we profiled, and then each row is one of these sarbecavirus RBDs. And basically this heat map colors black any antibody that does bind with high affinity to a given sarbecavirus RBD, or in white if it escapes. And so we can see, obviously, there's a lot of variation where many of these antibodies, particularly in the core RBD, show some of this breadth of binding to viruses from outside the SARS-CoV-2 clade, or even some of these bat clades from Africa, Europe, and this distant Asia clade. Whereas other epitopes are pretty restricted in their breadth, really only binding perhaps SARS-CoV-2 and the most similar RBD from one of these pangolin coronaviruses. So to kind of make sense of how this breadth is influenced by the details of particular epitopes, what we did next was just use this layout of epitope space generated here, but now project functional properties as these color scales onto this map. And so what I'm showing here is a quantification of that breadth that I just showed, mapped to each antibody in this space, where dark antibodies, such as S2H97, the broadest antibody we profiled, are this deep purple color, whereas the antibodies that have the least degree of sarbecavirus breadth are represented in yellow. And so when we project breadth onto the space like this, we can see pretty clearly the partition between broad antibodies being the ones down here that are these core RBD epitopes, whereas with a little bit of fine-grained resolution, some have a little deeper green than others. The antibodies that have less sarbecavirus breadth are again this RBM ACE2 contact surface. We can do the same thing now with our quantification of the frequency of natural escape mutants within the SARS-CoV-2 pandemic itself. And once again, we can see that these core RBD epitopes have generally a very low degree of variation among SARS-CoV-2, whereas some of these RBM epitopes are the ones where most of the sequence diversity is accumulating. But you can see kind of one interesting pattern, which is that there's a little bit more restriction about where the variation is happening in SARS-CoV-2 at this point compared to the breadth. So some of these antibodies that are beginning to have a little bit more breadth, perhaps, are correspondingly having fewer natural escape variants in the SARS-CoV-2 pandemic itself. And so to emphasize that here, I can take our panel of antibodies and just directly compare what is the degree of breadth that this antibody exhibits with respect to binding to all these sarbecavirus RBDs, where going to the right is a more broad antibody. And I can compare that to the frequency with which mutations that escape binding by that antibody are found in SARS-CoV-2. And so going down means there's fewer natural escape mutants for that antibody. And so you can see this kind of hockey stick shape where, once you get a certain degree of breadth, kind of this threshold between perhaps S2E12, which has that little moderate degree of breadth, which we'll go into a little bit more later, you begin to have greatly suppressed frequencies of natural escape mutants within the SARS-CoV-2 pandemic itself. And so this suggests that there's some degree of concordance on the types of evolutionary pressures that are causing variation to occur over long-term sarbecavirus evolution and the types of pressures that are causing variation within SARS-CoV-2 itself. And so to get a little bit more insight into kind of how and why these two scales of evolution might be linked, I think that can be shown if we walk through a couple detailed descriptions of a couple antibodies that really epitomize this relationship between natural escape and breadth. So I'll first focus on kind of the broadest antibodies from this sarbecavirus breadth perspective, which are these antibodies that bind within the core RBD, as opposed to those that bind in the RBM. And again, as I showed you before, the core RBD is where all of the antibodies that have some of this cross-clade breadth fall. And the reason for that is pretty simple, which is if we just look at the diversity and an alignment of sarbecavirus RBDs mapped onto the structure from more constrained positions to those where there's a lot more evolutionary variability, we can see that all of those positions with that evolutionary variability tend to map to the RBM or to the ACE2 contact surface, whereas this core RBD is more conserved. And so when we just map on the crystal structures of where all these antibodies bind, we can see that they're kind of decorating the surface of this core RBD where there's all of these generally conserved epitopes. And so, there's a couple different antibodies that bind further up or further down on this surface of the core RBD. Or if we sort of rotate this around to look at the backside, we can see antibodies like S309 are binding to more or less conserved surfaces on this other surface of the RBD. And so we can see these core RBD broad antibodies are really just decorating the surface of this core RBD. We were particularly interested in this S2H97 antibody because it really represented a new epitope class to us. We hadn't really seen antibodies that were binding this particular surface. And then correspondingly, we saw that it has the most remarkable breadth of these antibodies. So this is this antibody that's binding across all 4 clades of these sarbecaviruses, including even this orange clade, which I think differs at up to 40% of positions compared to SARS-CoV-2. So S2H97's epitope is pretty interesting. People talk a lot about cryptic epitopes in the RBD, which are those epitopes that are buried when RBDs are packed down in the spike trimer. And so S2H97 is almost like a supercryptic epitope, which I'll show here. So Nadine and Georgie at Vir solved the crystal structure of S2H97 bound to the RBD, which I'm showing here. And I'm also layering on our escape mapping, once again, showing that we picked up the structural epitope. But if we look at where this epitope is in the kind of closed spike trimer, we can see that the surface is deeply occluded by packing with the N-terminal domain or the NTD of a neighboring spike protomer. So as a consequence, it takes a lot of so-called opening of the RBD in order to expose this S2H97 epitope. So Young and David Wiesler at UW used cryo-EM to sort of visualize how S2H97 Fab is able to bind to this cryptic epitope. And so what I'm showing here is a structure that they refined that has 2 S2H97 Fabs bound to these open RBDs. And so in yellow is one example of an S2H97 bound RBD protomer, which shows the degree to which this RBD has to open and kind of bend upward in order to enable access to this Fab. And we can compare that to epitope targeted by this other antibody, S2A4, which is kind of the more classic cryptic epitope that a bunch of folks have worked on, where we see that the RBD has to open, but to a much less degree than is required for S2H97. And so this epitope targeted by S2H97, as I said, is highly, very difficult to access. And right now we're in the process of collecting a lot of functional and biochemical data on this antibody. But sort of as you'd expect from this type of structure, what we're seeing is that the way that it neutralizes is indeed by requiring this opening of the RBD and triggering those conformational changes to the post-fusion form. The last thing I sort of want to emphasize is that these S2H97 super broad antibodies are incredibly rare in polyclonal sera. So I'm showing here some data from Luca Piccoli at Vir, in which he performs these blockade, sorry, I'm missing the y-axis, these blockade of binding assays in which he takes sera, in this case from SARS-CoV-2 convalescent patients, and asks what dilution of sera is capable of competing off binding of each of these monoclonal antibodies across these different epitope classes. And so something that's higher up means you could dilute out the serum further and still compete off binding from that antibody, which implies that there's more of the antibodies like that antibody in that sera sample. And so when we look at the 4 RBM antibody epitope classes, we can see, there's a lot of activity of antibodies like these in these sera samples compared to the different epitope classes within the core RBD. And in particular, the S2H97 class, we can see that for the majority of these convalescent sera samples, there's no measurable competitive inhibition of S2H97 in these sera, suggesting that these antibodies are incredibly rare in these samples. And I think that's interesting. It in part explains why we haven't seen many of these antibodies. I think it also explains why there's not really variation at this epitope, perhaps over short-term evolution because there's no immune pressure. But I would suspect that this inaccessibility is probably true over long-term evolution as well. I think kind of one interesting future direction is that some folks are working on RBD-based vaccines, particularly in the context of nanoparticles, which might unmask epitopes like S2H97 and these other cryptic core RBD epitopes. And so you might actually be able to get more broad vaccine responses by directing activity towards epitopes like this. All right, in the last little bit, I'll kind of focus on one other epitope that we thought was quite interesting. This is kind of now focusing in on the RBM classes of epitopes, whereas I mentioned most of them have very limited breadth, only binding to the GD pangolin coronavirus RBD. But we did see one RBM antibody, which we call S2E12, which exhibits a little bit more modest breadth. So binding with breadth out to more like RATG13 and GX pangolin, which exhibit a little bit more divergence from the SARS-CoV-2 RBD than this GD pangolin. So just to confirm that little degree of breadth, Ale Tortorici and David Wiesler's lab performed pseudovirus neutralization experiments where she did confirm that S2E12 can indeed neutralize all 4 of these SARS-CoV-2 clade spike viral variants, although of course, as we see here, it does not exhibit breadth across to the green SARS-CoV-1 clade. When we look at the epitope of the S2E12 antibody compared to some of these more escapable RBM epitopes, we see kind of 2 key features. As I mentioned before, it has a low degree of escape mutations at its positions in ongoing SARS-CoV-2 evolution. And then kind of corresponding with that, we see a lot more of these yellow letters. So these letters that are that suggest there's deleterious effects of mutations within this epitope with respect to the basic function of receptor binding. So first, to kind of more directly show that S2E12 is more robust to ongoing SARS-CoV-2 evolution, Dora Pinto at Vir performed a host of neutralization assays with a bunch of the well-known and some of the lesser-known variants that have mutations within some of these epitopes. She saw indeed that S2E12 was capable of potent neutralization of all of these variants of concern, including many of the ones that have E484K, although sometimes in certain contexts, some of these variants had 3 or 4-fold decreases in neutralization potency, which is still relatively small considering some of these other escape mutations like E484K with S2D106 might have a greater than 1,000-fold decrease in potency. The other really exciting thing that we saw about the S2E12 epitope was sort of confirming that it is indeed functionally constrained as suggested by our ACE2 binding overlay. And so Zhoming Liu and Sean Whelan's lab performed some more of these viral escape selection experiments to see what types of mutants emerge when you grow virus in the context of S2E12. And so I'm representing that data here where we're showing all the possible mutations with respect to their effects on ACE2 binding on the y-axis. So going down means more deleterious. And on the x-axis is the mutation's effect on antibody escape. So further to the right means stronger escape from antibody binding. And then in the red diamonds, I'm illustrating those mutations that Zhoming got to grow out in this viral escape selection. And so you can see in general, a lot of these viral escape mutations are out here to the right, indicating that we measured they escape antibody binding. But for things like S2D106 and S2X58, we see, first of all, these mutations from variants of concern, but we also see that the mutations that are accumulating don't really have these deleterious side effects for ACE2 binding. Whereas with S2E12, it was sort of in this pinch where it could either take a mutation G476S, which we confirmed only had like a 4-fold effect on neutralization, but is fine with respect to receptor binding, or it could take these large effect antibody escape mutations that suffered some more moderate or severe decreases in ACE2 binding affinity. And to sort of confirm that this decrease in ACE2 binding affinity does matter for viral replication, Zhou Ming performed these competitive replication assays where he combined the wild type spike and these 4 escape mutants in the absence of S2E12 and just passaged them over time and looked at how they competed or changed in frequency. And he saw that this G476S mutation, which didn't have a deficit, but also wasn't that strong of an antibody escape mutation, basically grew as well as wild type. But these 3 mutations that emerged with stronger antibody escape suffered these replication defects of moderate effect for these 2 mutations or strong for this A475D mutation. And so we think this really confirms that the S2E12 epitope is functionally constrained, even though it's in this highly diverse RBM epitope class. Just to really emphasize that this is a super fine-grained detail that apparently can matter a lot for viral evolution. I'm comparing here structures of S2E12 versus S2D106 bound to the RBD, and just emphasizing that really it's about a 2 amino acid translation in really the focal interaction of these 2 different epitopes, where S2E12 really gloms onto this F486 residue, whereas E484K is kind of more up at the boundary of the epitope, and so it doesn't have as strong of an effect on binding. Whereas S2D106, as I said, is just translated over slightly to focus on this E484 residue. And so even though it's a minor structural shift, the difference in functional constraint and evolvability of these 2 positions is so extreme that this has big implications for viral escape. So I know that was probably kind of a lot, and so I kind of wanted to just finish thinking, kind of blending together some of the takeaways that we've been seeing, and then how that's leading to some of the future directions that we're thinking about, as well as a lot of other people are thinking about. So in this kind of third part of the talk, the thing that really surprised us was seeing that breadth of binding to sarbecavirus RBDs seemed to correspond to or correlate with robustness to escape among SARS-CoV-2 viral variants themselves. We think this can kind of emerge for 2 major reasons. The first is that there can be some sharing of the types of constraints that limit sequence diversity within the short-term SARS-CoV-2's evolution, as well as maybe more of the moderate or long-term evolution of sarbecaviruses. And perhaps that's the reason why S2E12's epitope isn't really changing in the SARS-CoV-2 pandemic, but also maybe has been conserved a little bit more over this moderate evolutionary distance to things like the GX pangolin virus. Perhaps another reason that could contribute to some of these shared constraints or shared phenotypes is that it's possible that there's also a shared lack of immune pressure over SARS-CoV-2 and sarbecavirus evolution. So it might be that in bats, where we do see a lot of variation at common antibody sites, epitopes like S2H97, which are just so cryptic, there hasn't really been any pressure on that epitope to evolve. So in addition, potentially to any constraint, there's also just not any positive selective pressure for evolution to be occurring at that epitope. And so, it's conserved both within SARS-CoV-2 as well as all the way out to the most distant viruses in bats. And so we think some of this insight about how breadth relates between these 2 scales can be incorporated into antibody and vaccine development. So a lot of people are working on kind of broader next generation vaccines, which have broader pan-sarbecavirus activity. And I think kind of fleshing out the different epitopes by which this broad activity can emerge can help in the evaluation and design of these vaccines. And in particular within antibodies, I think sort of highlighting what epitopes are more broad and also which ones are not as evolving under positive selection because of immune pressure can help us to kind of come up again with an arsenal of antibodies that targets more diverse epitopes and is more robust to ongoing SARS-CoV-2 evolution. Another important theme that I think we're thinking a lot about is how these different epitopes differ in their immunodominance. And by that, I mean the degree to which they're targeted within polyclonal sera, which is going to continue driving evolution of SARS-CoV-2 RBD. And so in some of Ali's early work, we've seen that polyclonal responses to the ancestral, the Wuhan Q1 RBD or spike is really dominated by antibodies to this E484 epitope. So antibodies like LY-CoV555 or S2D106. And at this point, there are other RBM epitopes, such as the one targeted by Regeneron 10933 and S2E12, that seemingly aren't yet driving the same degree of antigenic variation. However, I think it's going to be incredibly important to continue to pay attention to how immunodominance is going to shift as SARS-CoV-2 continues to evolve. And I think kind of rationale for this concern emerges from some of the observations about B1351, which looks really interesting. In that people have, so some work from Penny Moore and Tulio de Oliveira and Alex Siegel has shown that B1351, which incorporates the mutation E484K, sera elicited by this variant actually back neutralizes the prototype spike better than the ancestral sera neutralizes B1351. And so what that suggests is that perhaps this E484K mutation doesn't just simply replace K484 as the new immunodominant position, but instead it might be shifting immunodominance to other sites that are shared between these two different spike variants. And so, if more of the variants like B1351 begin to evolve and increase in frequency over longer term progression of this pandemic, it might be that some of these other epitopes that currently aren't driven by strong immune pressure begin to become more important and will also begin to degrade over time. And so that kind of brings me to this idea about the capacity for sort of medium and long-term antigenic evolution within the RBD. And so I think one thing we've been seeing in a lot of our work is that just in general, the RBD is broadly tolerant of mutations. So there's, from a biochemical perspective, a lot of capacity for this RBD to keep evolving through sequence space over time in many different directions. And I think that's echoed over sarbecavirus evolution as well, where this RBD is among the most variable regions of the entire virus. We've also now finally been getting information from our lab and others that other human coronaviruses, like these common cold coronaviruses, are indeed also evolving antigenically over time. I think there were a lot of questions about whether coronaviruses do evolve antigenically like flu or whether they're more like measles, but we're beginning to see that indeed these coronaviruses do evolve over time. And so I think this type of perspective mapping of immune escape that we're doing is going to continue to be important for informing development and application of antibody therapeutics and vaccines. And then the last kind of thing that I'm working on primarily right now is kind of taking this broader step back to sarbecaviruses writ large to kind of do these broader surveys about, what are the other zoonotic potentially zoonotic lineages within these SARS-related coronavirus reservoirs, and how should we be thinking about therapeutics and vaccines that might be more broadly protective against, these possible SARS-CoV-3s and so on. So with that, I'll thank, as I did at the beginning, Jesse and Allie, as well as a bunch of other people in the lab, a lot of really important collaborators. That's been one of the most fun bits of this work. So from David Wiesler's lab, James Crow's group, some of these scientists at Beer who have been incredible to work with, Sean Whelan's group, Jonathan Lee, who I didn't get to talk about that part of the work, as well as Neil King and Eric Mattson and funding. And thanks to everybody for chiming in. And I'm excited to chat about any of your questions.
Awesome, Tyler. That was amazing. Lots of great data, and you raised a mere 20 big questions for the field moving forward there.
Well, they'll be answered probably tomorrow at the current pace. So,
yeah. Let me. I just want to springboard off of something that you got to right at the end of your big picture questions. Have you guys looked at viruses slightly outside of the sarbecavirus family, like maybe the Murbecavirus?
Yeah, not really in a lot of detail. We have just a small amount of data that, S2H97 is like the broadest RBD antibody we've seen of all. And we do see in ELISA that it doesn't extend out to some of these other coronaviruses. And so, yeah, the idea about I think I mostly talk about pan-sarbecavirus, but this idea about pan-coronavirus is also a super important idea. And so I don't think, there might be more broad epitopes in the RBD that are still to be found, but I'm guessing that just the RBD is just too distinct between some of these broader lineages of coronaviruses, and maybe more of that pan-coronavirus potential is going to be more of these like fusion peptides and other domains of spike. to get that really, really deep breadth.
Yeah, or maybe not even spike. I don't know, like maybe nucleocapsid. I don't.
Yeah.
But then converse to that question, that one that really freaks me out a little bit is seasonal cold coronavirus NL63, which is an alpha coronavirus, but then binds the same ACE2 receptor again. So is that? I don't think there's a structure of that complex, is there?
I think there is. Yeah, I've because I know the epitope is known, so I assume that comes from structure. And actually, I've seen a structure, at least of the RBD of NL63. So it's definitely convergent. It binds the same overall surface of ACE2, but like if sarbecaviruses bind here, NL63 is like here. It's shifted a little bit. And the RBD of NL63, it has a similar looking folded core. Like I think all the coronaviruses I've at least looked at, which is, I haven't dug in that deep yet. They all have this similar beta sheet folded core. Then the RBM looks different on a lot of these. So like NL63, I think has, the RBM of SARS-CoV-2 is basically just like two loops, but NL63 has like almost a whole nother, like what looks like a domain there even. So it's definitely a very distinct at, the structural homology as well as the actual interaction interface. But yeah, I think like, why ACE2, is it just like a sitting duck, or like, is it the right tissues? That's, beyond my biochemical wheelhouse, but I think it's interesting.
Yeah, I mean, if I was a virus, like it seems like a really dumb target. I mean, it's super low abundance, like,
Yeah.
Better,
True. Yeah, I don't know.
Sarada, you want to hop in? And audience, please raise your hand, type your question in the box, etc.
Sure. Thanks, Taylor. So first of all, this is regarding the evolution of the virus. So there is no doubt that this virus will evolve. And as the RBD keeps evolving in the sequence space, do you think that this virus will accumulate enough mutations and at a rate fast enough for us to keep updating the vaccines? Given that the mRNA platform is quite amenable to updates, frequent update. Do you think you're looking at a situation like the flu vaccine where, at a seasonal or at a regular interval, we might need an updated version?
Yeah. I think that's kind of where things are headed. One good thing, some of these variants, we do know they're degrading vaccine-induced immunity, but it's still mild, right? It's like 4, 6, 8-fold. And these vaccines are so potent to begin with that like you can take that hit, and sure, there might be more breakthrough infections. We're seeing some of those reports, but it's not like, everyone's susceptible to these new variants. And so, surely more changes will continue to accrue. And then eventually, I don't know what the threshold will be for this virus to say, what time to try and update. But I don't see why those types of updates wouldn't be able to be quickly done, given that, the phase 1-2 trials for some of these new B1351 based spike vaccines are already happening with these mRNA platforms and things like that. So yeah, I think, I don't know what the time scale will be like. From a paper from Jesse's group on 229E, some of his data suggested that it takes maybe 5 years for viral evolution to escape a time point's polyclonal immune response to a substantial degree. But that's harder to compare different evolutionary dynamics up. So who knows what the time scale will be? But
yeah. I think like the real card, seems to be evolution in immunocompromised patients, where it can rip out 40 mutations in 3 weeks.
I
underlie a lot of the variants of concern.
Yeah, or at least they're pre-warning us about what the mutations are. It's hard to say where the variants of concern come from, but they're a lot of the same mutations. And I ended up having to hide those slides, but we saw a really interesting example with the Regeneron cocktail. And I feel like it's a really important kind of microcosm for how these antibodies are used globally as well, this was a paper that first came out from Jonathan Lee at Brigham Women's. And he had this persistently infected patient who was immunocompromised, who was infected for like 150 days with several bouts of viral spikes. And so for the first 145 days, all of his treatments were remdesivir and convalescent sera. So he was getting this like low level polyclonal immune selection, kind of population type immune selection. And then what happened was at day 145, they applied the Regeneron CoV-2 cocktail. But basically, when they did deep sequencing of this patient, they were already seeing a lot of low-level mutations kind of rising and falling through these first 145 days, such that when Regeneron-CoV-2 was applied, all of the like underlying variation was already there, where there were already haplotypes that had mutations that escaped both of these antibodies that just quickly took over the next time point after the cocktail administration. And so I think it's kind of a microcosm for the problem or the difficulty with these antibodies in the world, which is that, you know, the world's immunity is exerting pressure on this virus such that when we get to a patient who needs the monoclonal antibody, some of that variation is already happened. That renders some of these antibodies, you know, less potent or perhaps completely escaped. Yeah.
Mm-hmm.
So adding to that, I mean, we talked about vaccines, but sounds like the antibody cocktails available would also probably need an update based on, I mean, right?
Yeah. I mean, I think, you know, like bamlanivimab is already gone as a monoclonal, and even its cocktail is, you know, there's already variants out there that escape both components of that cocktail. And so mixing and matching it with other antibodies, or kind of my idea is, you know, if we have 6 different cocktails that have orthogonal sets of sites, you're ready to adapt to whatever kind of variant you might be expecting to see. But especially if we can get a lot of these epitopes that aren't changing as quickly, some of those cocktails will last longer as SARS-CoV-2 continues to evolve. Because as I said, I'm thinking, you know, 5-10 years, this virus is going to be there, and it'd be nice to not have to develop new antibodies every 6 months.
No.
I mean, and bamlanivimab may come back in style, you know, just
True.
like the antibiotics, like, you know,
True. Yeah.
If the escape, you know, mutations drift far enough, sometimes the first genuines start to work again. So
yeah, exactly. Like if K484 does begin to then become highly targeted by immune selection, it might just want to revert back to E. Yeah.
Yeah. Hey, I wanted to ask you more about vaccine design. You know, you showed that you're collaborating with Neil King. So, you know, I'm guessing that it's going to be like nanoparticle display of, you know, multivalent vaccines. Are you guys trying to, I don't know, come up with spike mutants that would tile the antigenic space, and then you would use that as like a polyvalent vaccine, or what's the idea?
You know, we haven't talked about mutants like that. We've talked more about like what types of Sarbeca viruses might you want to combine to get the types of epitopes that are more robust. And so like I took a lot of inspiration from that S2E12 antibody, which to me suggested like if you can bind with breadth to GX pangolin, for example, that's not incredible breadth, but it seems to be enough to at least be durable to the last year of SARS-CoV-2 evolution. And so it's maybe we don't need a pan-Sarbeca virus vaccine, at least yet. Like maybe if you have GX pangolin and SARS-CoV-2 in a cocktail, that will at least knock out the most variable of the variable epitopes and get a little bit more directed activity toward ones that are a little bit more robust.
But I mean, do you think that's the best bang for your buck? Like using like other naturally evolved ones like bat and pangolin viruses? I mean,
yeah, I mean, that's a good,
got the saturation mutagenesis data. Like,
yeah, no, that's a good point. Yeah, and we have talked about it. Like for example, like one of the simplest things we've seen is like we know a bunch of different places you can introduce glycans and not really mess up the RBD too high, and that's an easy way to mask epitopes. So we have talked about that, and you know, I think maybe we are getting to the point now where, you know, we're starting to see what that epitope space looks like, and maybe can say, you know what, I do want more of these antibodies. Like, let's try to hide these other things and get more of these. I think, you know, up until recently, I think my response has more often been like, well, we don't know enough yet about like what's good, right? But I think maybe at this point we are beginning to know that. It's a good question. And you know, we are kind of in a similar vein. We're working on a project now which would enable we basically took like our 18 most representative knockout mutations. So like we know E484K knocks out all these antibodies, and F486K knocks out all these, like, so we have these like 18 representative like charge introduction type knockout mutations, which could be useful in like designing probes to pull down certain antibody classes, or similarly, it could be to be like, well, let's prime with RBD, but then let's boost where we knocked out these 17 epitopes to beef up the response to this one epitope. So yeah, I think there's a lot of information out there that could definitely guide designed like that. Yeah, it's interesting.
Super exciting. All right. Well, Tyler, that was amazing. Yeah, thanks for your time. Thanks for sticking around to answer our questions. Yeah, best of luck. And
Thanks.
Matt, can you please bring up next week's slide? Yes, Dr. Peter Police. Next week, this will be a follow-on to Jason McClellan's talk last week, taking the polyproline designed stable antigens and then deploying them in a relatively new vaccine format to make stable, potentially low-cost vaccines that could serve most of the world. So don't miss it. It'll be super exciting. Okay, everyone stay safe. See you next week. Bye-bye.